Hi, my name is Viknish. I'm the founder and CEO of Cellivate Technologies. So a little bit background of myself. I did my undergrad at the State University of New York, Stony Brook. The nice thing about the undergrad education I got is that it really instilled in me the need to go deeper into what I was doing. I did my undergrad in biomedical engineering, and I realized that the undergrad education itself was quite superficial, and I wanted to go deeper into different areas. So as I was looking at different grad schools, I got a good package from the National University of Singapore. So I came back to Singapore, pursued my PhD here in biomedical engineering. So what inspired me in science? I've always been inspired in science. In fact, as a student, very often I'll do badly in every subject except for math and science. I generally have an interest, I have a passion for math and science. From a very young age, I knew I wanted to become a scientist. The reason I chose biomedical engineering was because it encompassed the different sciences, physics, chemistry, biology, and it also had engineering principles. So I felt it had components of everything I was interested in, which is why I pursued biomedical engineering and the sciences in general. If I were to go back even further, growing up, I was always weaker with the languages. So I was weaker with literature, with history, with English definitely, my second language for sure. It seemed like it took a lot of effort to do those subjects, but with math and science, it was more intuitive, it was more logical, and so naturally I found an affinity to the sciences. So during my PhD, my focus was on stem cell differentiation. During that time, I developed a system to differentiate neural stem cells into mature neurons, and the goal was for the treatment of different neurodegenerative diseases such as Parkinson's disease. After I finished my PhD, I was working on a project where we were trying to quantify sensation. This was really for cosmetic applications where the field was moving away from animal testing. So what we were doing is we used stem cells, differentiated stem cells to skin cells on one side and to peripheral nerve fibers on the other side, and we'd have these peripheral nerve fibers interact with the skin cells, and we would stimulate the skin cells with known allergens to try to measure a response on the nerve side. So as we were doing this study, one of the biggest bottlenecks we faced is that cells kept peeling off. We tried different commercially available methods such as collagen coating, fibronectin coating to have these cells attach better, but what we noticed is that due to the long duration of the experiment, cells tend to peel off, especially when done on glass, they tend to peel off, and we were not able to complete the study. So we started looking for alternative methods to have cells to attach better and even accelerate their growth. One thing led to another. We started a collaboration with a physics institute in NUS. The name of the institute was NUSNI, NUS Nanoscience and Nanotechnology Institute. In collaboration with them, we developed a surface coating that allowed cells to attach better and accelerate their growth. So once that study was completed, we published that work. We realized that what we had developed is more powerful than the study itself. And so I joined that physics institute and continued development of this thin film nano coatings that could help cells attach better and accelerate their growth.
Cell technologies is a big and growing field with many practical applications. Growing cells, or cell culture, is a lot like farming. You need the right conditions to get a good yield. For most cells, cell culture requires the adhesion of cells to the proper substrate before they can proliferate to larger numbers and differentiate into the cell of choice. Unfortunately, many cells don't adhere properly, produce low yield, and may even have unwanted mutations. These factors, together with the expensive cell media, drives up the cost of production. However, We have made a breakthrough in cell technology. We've developed and patented the use of a revolutionary coating that allows higher cell adhesion, lets cells grow at least 2 times faster, and unlike biological or chemical coatings, have a very long shelf life. This drastically reduces cost of production, increases cell yield, and thus profit for most applications. This method is also customizable to different cell types and is compatible with present-day solutions. Our products will be used with different types of critical cells so as to overcome many global challenges, from medicine to food.
It was around this time when the whole cultured meat bus was moving around, even in Singapore. And from talking to different companies, we realized that one of the bottlenecks they faced was the same bottleneck we were solving with our nano coatings, because many of these companies were having the problem of growing critical cells. Since these cells were taken out of the animal, these critical cells that would go on to become the meat of choice was having difficulty growing. So that's where we realized that our nano coatings could actually play a part, and that was really the inception of Cellivate Technologies. So when we talk about cultured meat, there are different parts to the process. The first part is to get a small piece of biopsy from the animal itself without slaughtering the animal. After that, critical cells are taken from this biopsy that would eventually become meat. Naturally, when we talk about meat, it's a composite of fat and muscle and other tissue. Cells of these types would be taken that would eventually become meat. Now, there are many ways to do this. We could take stem cells, or we could take adult cells, right? So I won't go into the detail of it. But the essence is that the cells are taken and critical cells need to be grown. Once we have a starting population of these critical cells, these cells need to be scaled up to very large numbers. So we are talking about trillions and trillions of cells to get a decent sized piece of meat, right? So not only do we need a lot of cells, we also need the cells to be structured in a certain way to make meat as we know it. So as you can see, there are different parts to this. Now, there are complexities involved in the types of cells. There are complexities involved structuring. There are complexities involved in the scaling up of cells. So all of these parts are quite intricate. But in essence, these are kind of the steps involved. Now, in our first innovation, where we discovered a nanofilm that allowed cells to attach better and accelerate their growth, we were able to have the critical cells to grow well, to have a starting population of critical cells. So in our second innovation, we came up with edible microcarriers to help these cells scale up into a very large number that can eventually be made into meat as we know it. Now, let me give you a little bit of background as to why this is important. When we grow cells, the number of cells is directly proportionate to the surface area we have. So a flask is about 75 centimeters square, and so you get a surface of 75 centimeters square that cells can grow. So if you have a larger surface, then you have more cells to grow. Now, when we are talking about billions and trillions of cells, then we need very, very large surfaces. Realistically, it's not possible to have that kind of real estate. So what biotech and pharma space, what they do is that they have these things called microcarriers. So these microcarriers are tiny beads that the cells can grow onto. And now, because these microcarriers are so small, and because we have so many of them, they sum up to have a very large surface area. For example, with some microcarriers, just a few grams of microcarriers could give us about 10,000 centimeters square of surface area. So that's really the logic. You have these very tiny beads, a lot of them in a small space that make up a large surface area. So in the pharma and the biotech space, these don't have to be edible, because after the cells are grown, the cells are extracted from these beads itself. But in the food space, these need to be edible for two reasons. If they are non-edible, then these cells need to be extracted out, which is an extra step. But when we talk about kilograms of meat, there's a very high risk of having these beads get stuck among the cells that are collected or among the kilograms of cells of meat. And if these are toxic, or rather if these are non-food grade, then it poses a food hazard. So for these reasons, our second innovation was on developing edible microcarriers. So these can actually be part of the food that people can eventually be made into the piece of meat. And then our third innovation is on having three-dimensional meat. So we all know that meat is a three-dimensional structure, and so our third innovation is on the self-assembly of three-dimensional meat that can be scalable. So this innovation we are working on will be meat agnostic. So it could be fish, it could be lamb, it could be beef, it doesn't matter. It would be a platform that allows us to have 3D meat in a scalable way. Doing research is very different from really making a product, because when we do research, we're trying to uncover something that is unknown. So that there's a certain system to do this, and it's actually not very difficult to do, because there's a lot of things that's unknown, and when we want to cover something that's unknown, it's honestly not very difficult to do. If you want to make groundbreaking research, that's a different story. But nevertheless, it boils down to covering or uncovering something that is otherwise not known. But when it comes to making a product, it's a whole different ball game, and it's a lot more complex, because first and foremost, there has to be a need for the product. So it could be very unique, it could be based on groundbreaking technologies, but if there's no need for a product like that, there's no application, there's no market, so it's actually very different. So when as a postdoctoral scientist moving into this space, initially I was less aware of the complexities involved. I knew that I was trying to fulfill a market need. I saw that there was a market need, but I was unaware of many of the other parameters involved, the regulatory hurdles, the commercialization part of things. So all of these things were things that I had to learn as I became an entrepreneur. Most definitely there is a change in skill set. So now, as the founder and CEO of Cellivate, I spend less time in the lab. I'm still involved in discussions, but I definitely spend less time in the lab overseeing technological development or any kind of R&D that's happening. Rather, my entire perspective, entire vision is more holistic, where for everything that's happening in the lab, I need to find the equivalent in terms of customer need. I need to make sure that enough funding is there to do the work. I need to make sure that the resources, including manpower, are accounted for, so the scientists that work with me are able to get the job done. So it's a completely different ball game. But I have to say that the training we get as scientists is actually very good, because this training is actually translatable to what I'm doing now. Even though I'm not directly doing science, I completely understand what my scientists need, and in that way I'm able to provide and also understand the bottlenecks they go through and provide the solutions that they need that may not be within their scope as scientists. So even though it's very, very different, and to a large extent I actually miss being on the bench trying to tinker around different things, making different findings or making different discoveries. To some extent I miss that, but I find it more exciting where I am now, because I am working on different parts of the whole system, and at the end of the day I know that what I'm trying to do is actually going to be beneficial to my users, to my customers. So I think one of the first challenges I faced when starting Cellivate is coming out of the scientific mindset. So as an entrepreneur, as I mentioned earlier, we have to look at different parts of the puzzle. But as a scientist, I was very focused on the product, and the technology is optimized such that the product works the best. So it took me some time to overcome this, to realize that there are bigger parts of the puzzle that are necessary to work together in order to make the startup successful. So I think that was probably the first thing I needed to overcome, the mindset shift. After that is the handling of different stakeholders. So as a scientist, there are very few stakeholders we deal with, because most of the time, like in my case, I was working with cells. I was working with my immediate supervisor, and that was pretty much about it, right? So I was very much in a silo, working on different aspects of my discovery, of my publication, whatnot. But as an entrepreneur, we have to look at work with different stakeholders. This includes our staff. This includes our investors. This includes our advisors and our mentors. This includes our potential customers. So all of these things need to work together. Initially, trying to come up with a healthy way to work with everybody took a little bit of learning. In addition, we also had to keep the vision of the company in front, and we had to make sure that all of our stakeholders do not disturb the vision of the company. And sometimes when there was no synchrony, then it was our responsibility to educate the different stakeholders or to bring them in line with our vision in a way that kept everybody happy. So these are kind of the soft learnings that we had to do as an entrepreneur. Yeah, so words of advice, certainly. The thing is, I think the Singapore landscape or the way Singaporeans are brought up these days are very different from many of the other Asian countries where they focus on being doctors, lawyers, engineers. But I think now with the different initiatives around the country, there's a lot of encouragement in STEM fields. Very often, the mindset of being in the STEM fields is that you either become a lab tech or a technician or a scientist or a teacher. But this is very, very far from the truth. The reality is that there are many, many fields you can go to, and the kind of rigor and training you get in the STEM fields actually help you to go into other areas. Like I spoke to this investment banker who is now, I think, in his early 70s. So he told me that it is his training in science and engineering as an undergraduate and and in his early careers that actually helped him to get into finance. And he said that these fundamentals in science, in mathematics, in engineering gives you the kind of logical thinking which you otherwise don't get in the other fields. So I think if a person naturally is inclined in the different engineerings, in mathematics, in physics, chemistry, I think they should pursue it, right? They should not feel that there may not be a future. They should not feel that, but I don't want to be a scientist, so why am I getting into this? I think they should pursue their curiosities. They should pursue their interests, because the world is such that opportunities keep coming up. And one thing I've realized is that by doing something that you're not passionate about makes the whole process of education more difficult. Yeah, so these are some things that I can think of that I would say. Today I'm going to give you a brief tour of our lab space. We have two lab spaces. One of them is at the National University of Singapore. That's a smaller lab space where we do specific cell culture and where we have our bioreactor. This here is our main lab space. It's located at the German Center at Jurong East, Singapore. So let me show you more. Within the German Center is the Life Science Incubator where we are located. Let's go. This room is our fridge room where we store a lot of our temperature sensitive samples. So we have 4 degrees fridge, we have a -20 fridge and a -80 fridge. Down here we also have a liquid nitrogen container where we store a lot of our biological samples. This over here is a bioreactor facility. So eventually our bioreactor will be moved to this room. This here, the autoclave room, is where we sterilize our samples and our equipment before we use them. All of our imaging of our different samples, our microcarriers, of cells growing on microcarriers, our 3D samples, all of these are imaged using highly sophisticated microscopes. This is our tissue culture room where our cell culture is done. This here is our tissue culture hoods. These are our incubators that we use on a daily basis. Like I mentioned earlier, this is one of the places where we do cell culture. We have another cell culture facility in the university. Apart from cell culture, we also do a lot of material science work. Okay, that part is confidential, but I hope you enjoyed this brief tour of our laboratory space, and I invite you to definitely come and visit us in person. Thank you.